Hello, my name is Yidong. I am from Academic Magnet High School, and today I will be presenting our research on mitochondrial ER stress pathways as therapeutic targets in smoking-induced invertible disc degeneration. I'll go through an introduction, followed by the aim of the study and hypothesis. Then I'll explain the methods and results, and finally conclude with the key findings and future direction. First of all, why do we experience low back pain? Low back pain is the second most common reason for hospital visits worldwide, and for many, it becomes a constant companion. But what if the root cause isn't just a sprain or strain, but the gradual degeneration of the intervertebral discs in our spines? Intervertebral discs are located between each vertebrae and are essential for spinal flexibility and load distribution. The IVD is separated into three regions: the core nucleus pulposus NP, the outer annulus fibrosus AF, and the cartilaginous end plate CEP, which is the region we focused on. When the IVD degenerates, the NP dehydrates, the AF weakens, the CEP of the disc bone interface calcifies, and the disc height decreases. This breakdown of the disc triggers the growth of blood vessels and nerves into the disc, leading to inflammation and chronic pain. There exist several risk factors for IVD degeneration, including smoking, genetics, diabetes, and alcohol consumption. Among these, smoking is a primary factor. Smokers are nearly 4 times more likely to develop disc herniation and show 20% higher degeneration scores. They also tend to experience delayed recovery after surgery. Despite known risks, cigarette smoking remains a major health issue. In the US, 11.6% of adults are cigarette smokers. Additionally, the percentage of people who experience secondhand smoke exposure is exponentially larger. IVD degeneration has no cure. Even if you are diagnosed as early as 20 years old, doctors can usually only offer pain management options until spinal fusion surgery or disc replacement becomes necessary as the condition worsens. Since it remains incurable, more research is needed to prevent progression and find a solution that is able to target IVD degeneration earlier and more locally. Oxidative stress arises when the amount of reactive oxygen species exceeds the capacity of antioxidants to neutralize them, resulting in cellular and tissue damage. Cigarette smoke contains nicotine and other harmful chemicals which can increase ROS levels and cause oxidative stress in the cells. In our research last year, we used the same in vitro IVD organ explant culture system we developed and found that cigarette smoke exposure CSE increased oxidative stress, apoptosis, and calcification in IVD, as reflected by elevated ROS levels, calc-based-3 activities, and the ALP activity. We also observed that different regions of the IVD show varying levels of damage, with the CEP being the most susceptible to oxidative stress. Building on the findings, the goal of our current research is to determine how CSE-induced oxidative stress damages organelles and promotes CEP apoptosis and calcification, quantify how CEP calcification alters disc bone interface mechanics relevant to IVD degeneration, and explore early therapies to restore IVD function. We hypothesize that CSE induces mitochondrial dysfunction and ER stress with the unfolded protein responses in CEP cells, which promotes a procalcific phenotype. This calcific transition disrupts disc bone interface mechanics and contributes to IVD degeneration. Organelle-targeted redox therapy may restore cell function and mitigate CSE-induced IVD degeneration. Next, I'll present the methods and results. Here's a brief overview of the study design. First, we prepared and quality checked the CSE. Then we isolated rat IVDs, cultured them as explants, treated them with CSE, and dissected the CEP tissue for proteomic analysis. We also isolated CEP cells, cultured them, and treated them with CSE for functional studies. Finally, we used computational models to examine how CEP calcification affects disc bone interface mechanics. I'll go into details on the upcoming slides. Let's start with the preparation of the CSE. Research-grade cigarettes were used for this process. A syringe was used to draw 35 milliliters of smoke over 2 seconds. As the syringe was pulled, the mainstream smoke bubbled through the culture media in a falcon tube, mimicking the act of inhaling smoke. Each puff was followed by a 28-second rest interval, and the smoke in the syringe was removed. Each cigarette was fully consumed after 14 to 15 puffs. This procedure produced a 100% CSE solution, which was then used for the subsequent experiment. We assessed the quality of the CSE using 2 methods. First, absorbance at 320 nanometers was measured to estimate the amount of organic compounds present. Second, the nitrite levels were measured using a grease reagent kit which produces a pink azo dye detectable at 548 nanometers. As shown in both graphs, the results were consistent with no significant differences between preparations, confirming that the CSE preparation was reliable and reproducible. Now let's move on to the IVD tissue experiment. Rat IVDs were randomly assigned to either a control group or a 10% CSE treated group. After 3 days of treatment, the discs were dissected into 3 regions: the MP, the AF, and the CEP. Rather than analyzing the entire IVD as we did in our previous research, we focused specifically on the CEP because it appeared to be the most vulnerable region. We prepared protein lysates and sent the samples for mass spectrometry to evaluate both oxidative protein modifications and changes in protein expression. The proteomic data were then analyzed using Metascape for pathway analysis, which helped us identify the major pathways affected by CSE treatment, including mitochondrial functions involved in oxidative phosphorylation. Our findings revealed extensive mitochondrial cysteine oxidation in the CEP following CSE treatment, particularly in proteins involved in mitochondrial respiration, including subunits of complexes 1, 3, 4, and 5, as shown in Figure A. Figure B shows that cysteine oxidation in these subunits increased by 28 to 65% after CSE treatment. These findings suggest that mitochondrial respiration is impaired, which may force CEP cells to rely more heavily on glycolysis to compensate for reduced energy production. Consistent with this mitochondrial damage, global proteomic analysis showed widespread protein changes in the CEP. As shown in Figure A, glycolytic enzymes were markedly upregulated, further supporting a metabolic shift toward glycolysis in response to mitochondrial dysfunction. Meanwhile, figures B and C show activation of ER stress regulation and intrinsic apoptosis pathways, indicating a shift toward a pro-apoptotic state in the CEP. In addition, proteins related to calcification, collagen cross-linking, and vesicle-mediated nucleation were altered, collectively supporting a pro-mineralizing CEP phenotype. To better understand the mechanism, we isolated CEP cells from rat IVDs, cultured them, and used functional assays to assess mitochondrial and ER stress pathways. We measured mitochondrial RS using mitosonks and mitochondrial membrane potential using TMRN. After CSE exposure, MTROS increased significantly, while mitochondrial membrane potential decreased, indicating mitochondrial injury. When we added NAC, a global antioxidant, both the ROS increase and the membrane potential loss were partially reversed, suggesting that this damage is at least partially driven by oxidative stress. We then assessed mitochondrial metabolism using the mitoplate S1 assay. After permeabilizing the CEP cells with saponin, we measured their ability to use pyruvate and other Krebs cycle substrates to further evaluate mitochondrial function. CSE markedly reduced substrate utilization, dropping to about 40 to 74% of control levels, and this pattern is consistent with broad mitochondrial dysfunction and impaired oxidative phosphorylation. We also examined overall cellular metabolism by measuring glucose consumption and lactate production in the culture medium. As expected, CSE-treated CEP cells showed increased glucose uptake in Figure A and increased lactate production in Figure B, as the cells have to rely more heavily on glycolysis to maintain energy production when mitochondrial function is impaired. NAC partially reversed this glycolytic shift, suggesting that oxidative stress contributes to this metabolic change. In parallel, we used real-time PCR to examine the major ER stress pathway to confirm that the trends observed in global proteomic analysis of IVD tissue were consistent with those seen in CEP cells. In response to oxidative stress, compared with controls, CSE-treated cells showed marked activation of the PERK, ATF4, and CHOP axis, indicating that CSE also induces ER stress-mediated apoptotic signaling in CEP cells. In this figure, we built a computational mesoscale model to simulate collagen fiber mechanics at the disc bone interface, especially in the CEP. The images show the anterior and posterior annulus fibrosis regions outlined by the red dashed lines. Based on these structural differences, we created 2 models, an angular model and a diameter gradient model. This allowed us to better understand how calcification changes the mechanical behavior of the disc bone interface. By adjusting the stiffness parameters, we simulated calcification in the interface. As calcification increased, the interface became stiffer and could tolerate higher stress, but it also failed in a more brittle way. In the graph, this is reflected by a steeper response and a sharper drop after peak stress, suggesting reduced flexibility and greater risk of sudden failure. In conclusion, our study suggests that smoking-related disc degeneration is largely driven by damage to the CEP. CSE induces ER stress and mitochondrial dysfunction, which promotes cell death and calcification in this region. As a result, the cartilaginous end plate loses its ability to properly support disc function at the disc bone interface. This highlights the CEP as an important therapeutic target for preventing or slowing disc degeneration. For our future direction, we plan to evaluate therapies that target specifically mitochondrial dysfunction and ER stress rather than a global antioxidant such as elamipretide for the mitochondria and 4-PVA for the ER. We also plan to move this work into preclinical studies and small-scale clinical trials. Ultimately, our goal is to work with pharmaceutical partners to develop locally injectable therapies that can preserve IVD function and slow disc degeneration so that doctors can provide medical solutions for early IVD degeneration diagnosis. Finally, I sincerely thank Dr. Wu for providing the resources and mentorship at MESC, his team for their invaluable guidance, and my science teacher at Academic High School for her continuous support and encouragement. And a special thanks to IYRC for organizing this event and giving me the opportunity to present. Thank you.